I'm an immunologist. I know that many of you are not, but what I say to the early stage scientists here, that this is a great time to be an immunologist, because what we're understanding is that alterations in the immune response or inflammatory response underlie not only the obvious autoimmune and infectious diseases, but a number of other chronic diseases that we hadn't associated with immunity, and that will involve neurodegeneration and mental health, type 2 diabetes, a lot of impact in cancer, atherosclerosis, and so on. So immunogenomics, I think, is very timely to think about the genomics of different diseases beyond just immunology diseases, but where there is an impact of the immune system. So we're interested in chronic inflammatory diseases, and of course these are complex, multifactorial diseases involving the interaction between genetics and environmental factors that play out amongst tissue cells and immune cells. So when there is a host maladaptation between the genetic landscape and these environmental drivers, this can lead to chronic inflammation in the gut in the form of inflammatory bowel disease, in the skin, rheumatoid arthritis, ankylosing spondylitis, and in the skin in psoriasis. And what we're understanding, and a theme that perhaps I'll come back to at the end, is that amongst these diseases there are common components, shared pathways that are involved in the pathogenesis, as well as some unique elements that perhaps relate to that interaction between immune cells and tissue cells. So it will, of course, be of interest as we think about therapeutic targets that might be relevant in one disease, how they may cross across different diseases based on the processes that they're targeting. Now, of course, an important component in health and the wiring and function of the immune system is our microbiome. We now understand that no matter how human we may feel, we are in fact a host microbial superorganism. Our body surfaces are colonized by a wide range of microbes, bacteria, viruses, and fungi. In the intestine is the largest density of bacteria on the planet. 100 trillion bacteria inhabit the human intestine, and the signature, the types of bacteria that are present are different between different individuals. And of course, these commensal bacteria contribute to not only the genetically, but also in terms of proteins that they make, and most importantly, the metabolites that they produce. And they contribute, of course, to our physiology in terms of nutrition, immune system development, and host defense. And when that interface is maladapted between the host and its commensal bacteria, and I'll be talking about this particularly in the setting of the intestine, this can promote disease. And rather like the immune system, we're finding that maladaptations of the host microbe interface are relevant across a range of different diseases. Of course, the challenge is to understand what is causative and what is correlative, and we therefore need to sort of develop models to be able to probe how the host microbe pathways function normally and how they can become deranged in disease. So as I mentioned to you, the gut is a primary site of host microbiota symbiosis. The bacteria here play an important role in promoting the right types of immune response to be able to drive barrier function and host defense. That involves a number of different bacteria that are being identified, often through the metabolites that they produce, impacting on the type of host response that is dominant, and driving key pathways such as secretion of mucins from the epithelial cells, antimicrobial peptides, innate pathways, and IgA. And what this does is promote this, what Laura Hooper beautifully described as the demilitarized zone. This is a staining here for bacteria in the lumen of the intestine. The blue marks the epithelial cell monolayer, and I think you can see that there are not very many bacteria growing in this part in this interface, and this is an important facet of compartmentalization and host defense, and therefore a healthy relationship with our intestinal microbiota. Now, what appears to happen in inflammatory bowel disease is that relationship breaks down. So inflammatory bowel disease is a chronic inflammatory disease of the gastrointestinal tract. There are different clinical forms, Crohn's disease and ulcerative colitis. To summarize what's going on in IBD, this is a chronic disease, it's debilitating, and it's increasing in incidence. Many patients will respond to frontline immune therapies in IBD, but a large proportion won't, and we currently have no way of predicting who will and who won't, and how to target therapies to the right patients in terms of the processes that become deranged. The sort of consensus of opinion is that there is a deranged response to the microbiota in genetically susceptible individuals. So here you have this interaction between genetic drivers and environmental drivers that can impact on the nature of the microbiome and the immune system, such that this is now maladapted, leading to the development of chronic intestinal inflammation. So it's an exciting time in inflammatory bowel disease research. We are now being able to appreciate results that have derived from the large genome-wide association studies that some of you, a number of you in this room have been involved in, results from the animal models that we and others have been working on, together with the capacity to be able to manipulate human immune cells in a way that wasn't possible. I think there are now very important iterative studies between mechanistic in vivo studies in mice, how you can manipulate the human immune system and relate this then to the genetic pathways that are being identified not only in genome-wide association studies in the polygenic inflammatory bowel disease situation, but also in monogenic diseases that probably Nadine has already mentioned to you, and of course provide a proof of principle in terms of key pathways that are involved in promoting intestinal homeostasis. So if one has a look at what has derived from both the study of polygenic as well as monogenic diseases that present with inflammatory bowel disease, we can map these genes into pathways that what we would consider identify key pillars of intestinal homeostasis. And this comes from a review that my colleague and myself, Holm Ulig and myself wrote last year. And the key pillars are, again, how we compartmentalize the microbiome. So appropriate barrier function, innate defense, mononuclear cell microbiocidal function. All of these working to make sure that we keep bacteria out. We can accept the goodies, but we don't want them coming onto our territory. And if they do, we have to gobble them up and kill them. And where there are deficiencies in these, in some of the immune deficiency diseases with alterations, for example, in reactive oxygen species generation, this can lead to chronic intestinal inflammation. The other pathway that stands out markedly, particularly from the monogenic diseases, is that when there is a dysregulation of the control mechanisms within the immune system that temper the inflammatory, the innate and adaptive inflammatory responses. And that relates to deficiencies in regulatory T cells. These are specialized FOXP3 expressing cells, production of interleukin-10. And when there are deficiencies in these, this can also, in monogenic diseases, give rise to inflammatory bowel disease. And I've blown this up here a little. This is very relevant to the pathways that we have identified from the mouse models, but monogenic loss of function in a number of pathways associated with the regulatory T cell axis and production of interleukin-10 drive inflammatory bowel disease in these monogenic diseases. it's also another aspect that is emerging and something that is limiting our ability to effectively use checkpoint blockade in various cancers, is that a number of the checkpoint blockade strategies will target the regulatory T-cell axis. And one of them is anti-CTLA-4, and this is an important pathway through which regulatory T-cells function, monoclonal antibodies that inhibit the function of CTLA-4, that promote anti-tumor immunity, will lead to the development of colitis in some patients. So these are also presenting an opportunity to follow the factors that are involved in why some individuals would develop colitis in this setting with anti-CTLA-4 and others would not. So a lot of opportunity in terms of mapping the genes into these different pathways and then understanding how we can influence the function of the regulatory T-cell axis to promote intestinal homeostasis. So our studies initially in mouse models were one of the early studies to identify the regulatory T-cell axis. We showed using a mouse model that transfer of naive T-cells into mice that have no T-cells of their own leads to the development of a chronic inflammatory response driven by the intestinal microbiota. If we transfer these T-cells into mice that do not have a replicating intestinal microbiome, then they don't develop disease. But when there are the microbes there that become targets of T-cell mediated and innate immune attack, you get a chronic inflammatory response. And we identified a number of cytokines that were elevated in that setting. One of those was TNF-alpha, and of course you know that anti-TNF-alpha antibodies are now one of the mainstay treatments for inflammatory bowel disease, but not all of the patients respond to that. The other cytokine that came out very strongly in these early model studies was the cytokine interleukin-23, and I'll be talking about that in a little bit more detail. If we transferred these regulatory T cells that are a specialized type of T cell that develop in the thymus and can traffic into the periphery where they can regulate immune responses, then regulatory cells were able to inhibit the development of intestinal inflammation in this model. And if we transferred them in a setting where the mice already had colitis, what you would have with an inflammatory bowel disease patient, these regulatory T cells could resolve the inflammatory response and cure the colitis in this model. And we showed that the ability of regulatory T cells to be able to do this was dependent on these cytokines, interleukin-10, TGF-beta, and the negative regulator of T-cell activation, CTLA-4, all pathways now identified in monogenic diseases that present with inflammatory bowel disease. And I think what this tells us is that mouse models are highly relevant, but we have to understand what spectrum of disease they are actually modeling. And what I suggest to you here is that this model, where there is a T-cell response in the absence of the normal regulatory counterpart, will represent some of these extreme forms of IBD, like you would find in FOXP3 deficiency or IL-10 deficiency. Of course, in the polygenic diseases, it's much more complicated, but we can think about how we can map the pathways that are relevant in monogenic diseases, perhaps into subsets of patients with polygenic disease. Now, the other aspect, another model that we have used that sort of reveals this point about the importance of host-derived interleukin-10 as a mechanism of mutualism and homeostasis in the intestine. This doesn't use immune-deficient mice. This uses wild-type mice, black 6 mice, infection with this mouse pathobiont, Helicobacter hepaticus, leads to the development of a chronic infection with Helicobacter hepaticus. But the mice live very happily with this bacteria because the regulatory T cells produce a lot of interleukin-10. And in fact, we and others are studying what's special about Helicobacter in being able to promote a regulatory T cell response and the production of interleukin-10. But if we now force a maladaptation between the host and its microbiome, now with the presence of Helicobacter hepaticus, this leads to chronic intestinal inflammation driven by the cytokine interleukin-23. And importantly, and I'll come back to this mouse model, although it responds to anti-IL-23, and I'll take you through some of the biology that we've been accumulating to help us understand how IL-23 is working, but it does not respond to anti-TNF. So it could be an interesting model for thinking about TNF-independent models of intestinal inflammation. So what I'd like to do this afternoon then is review some of our studies on IL-23, update you on its progress in the clinic as we think about how we can better identify the subsets of patients that would be responsive to anti-IL-23 blockade, and then cover a new pathway that we and colleagues have identified that involves the interaction between leukocytes and tissue stromal cells in the intestine, that we believe is an important driver of anti-TNF therapy resistance in inflammatory bowel disease. So interleukin-23 then is a common link in tissue inflammation. IL-23 is a heterodimeric cytokine composed of a P19, a unique P19 chain, and a common P40 chain that is shared between IL-23 and IL-12. It binds to a heterodimeric receptor composed of a unique IL-23 receptor chain and a common IL-12 receptor beta-1 chain, which is also a component of IL-12. And in fact, this crossover of subunits between IL-12 and IL-23 meant that for a long time investigators thought that immune pathological responses in tissues were driven by IL-12 because there were tools to target IL-12, primarily targeting these common chains, which when further tools became available, it was clear that IL-23 was having a more unique role in driving tissue inflammation. The pathway is increased in inflammatory bowel disease in terms of production of interleukin-23, and what's important here and of relevance to this audience, of course, is that multiple SNPs in genes involved in this pathway are associated with chronic inflammatory diseases, inflammatory bowel disease, ankylosing spondylitis, and psoriasis. And they're all targets for anti-IL-23 therapy in the clinic. So what does the IL-23 receptor pathway look like? I'm sort of highlighting it here in the context of the intestine, and it's quite clear that this pathway is relevant in intestinal homeostasis and host protective immunity, but can become deranged, leading to chronic intestinal inflammation. So what we know is that following intestinal damage, infection, or in the setting of impaired immune regulation, components of the microbiota will stimulate myeloid cells through their expression of pattern recognition receptors, and those myeloid cells, dendritic cells, and macrophages will produce interleukin-23, and this is prominent in the intestine, but varies depending on the nature of the microbial drivers. But compared to other sites, such as the spleen or lymph nodes, IL-23 is produced at higher amounts at barrier surfaces. So it's driven by the response to microbes, and when it's produced, it binds to its heterodimeric receptor. And the earliest biology on IL-23, of course, identified these so-called Th17 cells that would respond to IL-23 together with T-cell receptor ligation and produce a range of cytokines, particularly the IL-17 family cytokines, hence the name Th17. What we now know is that the IL-23 receptor is produced on a lot of different lymphoid populations. In addition to Th17 cells, it's expressed by a large range of unconventional lymphoid cells, including gamma delta cells and innate lymphoid cells that I'll be talking to you about. And this response can promote host defense for barrier integrity and neutrophil recruitment, but when it's sustained, it leads to this chronic inflammatory responses. So what we wanted to know is, how is IL-23 actually driving intestinal inflammation in these model settings that I've mentioned to you? And I'm going to just review some of the functional studies that identified a role for IL-23, one in promoting a particular type of inflammatory Th17 cell that produces high amounts of GM-CSF, as well as stimulating what at the time was a very poorly understood population of innate lymphoid cells that are also present at barrier surfaces, express the IL-23 receptor, and can be stimulated to produce a range of the same inflammatory cytokines as the adaptive Th17 population of cells. And alongside its ability to drive these pro-inflammatory responses, one of the reasons that targeting IL-23, I would say, is so efficient at inhibiting intestinal inflammation is because it also impedes the regulatory T-cell response. So if you take it away, not only do you reduce pro-inflammatory drivers, but you promote the regulatory T-cell axis. And I'll just show you some of that original data. Here is our transfer model of T-cells into mice that have no T-cells of their own, and that leads to chronic intestinal inflammation. If those T-cells don't express an IL-23 receptor, then they're not able to cause intestinal inflammation. This inflammatory population of Th17 cells that produce IL-17 and interferon gamma is markedly reduced, and the FOXP3 expressing population is increased. So it's influencing the balance of effector and regulatory T cells to promote an anti-inflammatory state. Now, one of the pathways that is increased markedly in a setting of chronic intestinal inflammation because of a regulatory T cell deficiency is the accumulation of a number of myeloid cells, including inflammatory monocytes and neutrophils. And that led us to ask if there is a role for cytokines that are involved in stimulating bone marrow hematopoiesis and the export of cells from the bone marrow that lead to an accumulation of mononuclear phagocytes and other myeloid populations. And indeed, in mice with colitis, there's an increase in a cytokine that plays an important role in driving this process, and there are subsets of T cells that are that involve production of high amounts of GM-CSF, as well as other cytokines associated with the Th17 response. And if we genetically manipulate the GM-CSF receptor by utilizing a knockout mouse lacking a chain of the GM-CSF receptor in this model of Helicobacter-driven colitis, now again with IL-10 blockade, what we find is that there is really virtually no intestinal inflammation in this setting. This is even more striking than taking away IL-23. So GM-CSF plays a very important role in IL-23 dependent colitis, and that is in part through influencing and promoting the accumulation of different populations of myeloid cells, including inflammatory monocytes, neutrophils and eosinophils in the intestine. So that led us then to identify this link between IL-23-driven intestinal inflammation and an increase in the GM-CSF axis, making us think about how cytokine pathways that are mediating inflammation in the gut are actually also talking systemically, particularly to the bone marrow. And I haven't gone through all of this data. We've published it, but it's in sort of a cartoon form here, when they're in the setting, again, of intestinal inflammation, where there is a lack of appropriate regulation because of either blockade of IL-10 or deficiencies of the FOXP3 expressing regulatory T-cell pathway. Now you have an accumulation of GM-CSF producing Th17 cells, and GM-CSF acts in the bone marrow to skew hematopoiesis towards these myeloid progenitors that exit into the tissue, promoting the accumulation of inflammatory myeloid cells of different flavors that mount a tissue toxic inflammatory response. And of course, this skew away from erythropoiesis towards myeloid progenitors may, in part, explain the chronic anemia that is present in a number of chronic inflammatory diseases. Is this to do with cytokine reprogramming of the hematopoietic response that then is becoming extremely deleterious and tissue toxic? Would something along here be a relevant new target? Well, GM-CSF, I think, is complicated in the context of the intestine, because GM-CSF also has an important role to play in intestinal epithelial cell barrier function. But for some other Th17 responses, it is being tried. There are currently trials going on with anti-GM-CSF in ankylosing spondylitis, for example. Now, these are the studies that led to the identification of these IL-23 responsive innate lymphoid cells, and they came from an observation made many years ago by Sophia Bonacori and colleagues when they were in the lab, and that involved the finding that rag mice that have no T cells of their own, there's no adaptive immune system in these recombinase activating gene deficient mice because they can't rearrange their T cell or B cell receptors, so they're not selected. So they are immune deficient and they don't have any T or B cells. If we infected those with this bacteria, Helicobacter hepaticus, this led to the development of an inflammatory bowel disease-like response. It led to chronic intestinal inflammation, and we could inhibit that intestinal inflammation by taking away a number of the cytokines that you would take away for T-cell dependent disease, but there weren't no T cells in these mice. And Sophia went on to identify a population, at that time a new population of innate lymphoid cells that express transcription factors related to the Th17 subset of T cells. These cells express the IL-23 receptor. They are prominent in the intestinal mucosa, and where in this setting, the bacteria stimulates a potent IL-23 response that drives the production of inflammatory cytokines and the development of colitis. And if we by antibody depletion studies, remove that population of innate lymphoid cells, that could both prevent and cure disease in this setting. Now, innate lymphoid cells over the last decade have been an enormous area of interest. They were originally identified, I guess, by Rainer Mabius, these raw gamma T expressing cells that were to do with lymphogenesis and involved in the development of lymph nodes and various lymphoid structures. But these studies and others by many colleagues around the world identified that ILCs, innate lymphoid cells, are not just relevant during the developmental phase, that there are populations in the periphery that have a role in microbial sensing primarily through the expression of cytokine receptors that they can then respond to and produce a range of inflammatory cytokines. So there are now a number of different innate lymphoid cell subsets reviewed by my colleague Jürgen Spitz, and these ILCs are very similar in terms of the genes that they express and some aspects of their function to T cells. There are some that mirror the Th17 population, some that mirror the Th1 population, and some that mirror the Th2 population. And there's evidence for these subsets of cells in terms of host defense, and also that they may become deranged in aspects of human disease. But of course, there's a lot of interest in understanding what might be unique to ILCs compared to what T cells are doing. Because what is clear is that these cells are very, very infrequent. They're very low amounts in tissues, although they may be in regional areas at high density. And of course, they seem to be able to confer a remarkable ability to regulate multiple physiological processes throughout the body, and this is taken by from a review by David Artis and Jürgen Spitz. And this just shows you at the various barrier surfaces in the body the presence of these innate lymphoid cells, which have been associated with different diseases. And I thought a lot of interest in understanding whether these may represent new targets or how we might harness them to promote intestinal barrier function. So in summary of this part, what we have postulated is that an important component of intestinal homeostasis and how the host responds to the microbiota is the balance between the production of interleukin-10 on one hand, driving pathways that are involved in intestinal homeostasis, and interleukin-23 and some other cytokines on the other. When there is a relevant microbial driver, and we have used the model of Helicobacter hepaticus, in the setting of a host maladaptation, where there are no regulatory T cells or no IL-10, this then drives this chronic IL-23 driven response with high amounts of GM-CSF and the elaboration and accumulation of a number of myeloid populations. And the IL-23, based on these and other studies, as well as the genetic studies, is of course a new target for inflammatory bowel disease, and these studies just come out in 2017 and 2018 are showing great promise in Crohn's disease, particularly anti-TNF resistant Crohn's disease. So this illustrates how understanding pathways at the cellular and molecular level in mouse models can then be translated into effective therapies. But I think the challenge is, although this shows great promise, we don't really know which patients to treat with anti-IL-23 or which ones to treat with anti-TNF. It is just worth mentioning that I have connected IL-23 with downstream cytokines such as IL-17, that IL-23/Th17 response that has been shown to be involved in a number of chronic inflammatory diseases. It was actually rather surprising to find that I've mentioned to you that anti-IL-23 works well in psoriasis and is showing great promise in Crohn's disease. It's really revolutionary in psoriasis. One treatment is sufficient to have long-lasting protection in really devastating forms of the disease. Now, anti-IL-17 also works well in psoriasis, but doesn't work in Crohn's disease. And that suggests that the mechanisms that are operating of this IL-23/Th17 pathway are not the same in all tissues. Hence, the commonality is IL-23, the unique component is IL-17 in the skin, but not in the gut. And there were some lovely studies trying to dissect this by Dan Kwa and Jen Town and colleagues, which identified that in the gut, IL-17 plays a very important role in barrier function, and it doesn't play that role in the skin. So if you take IL-17 away in the gut, then that is deleterious because it doesn't, the sort of compartmentalization and barrier function that I mentioned to you is being compromised, even if you're taking away IL-17. By contrast, IL-20, what it appears is that there are IL-23 independent sources of IL-17 in the gut. So if you take IL-23 away, you're not compromising that barrier function because you still have some IL-17. So again, this biology is explaining why things may work in one setting and not the other. So what I'm telling you is that there's a growing diversity of therapeutic options for inflammatory bowel disease. Our sort of toolbox is becoming quite well stocked, but it's very difficult for clinicians to know who to treat with what. There are antibodies that target TNF, IL-23, how T cells actually come in to the inflamed tissue. But I think developing strategies to predict therapeutic outcome will be critical for the delivery of individualized patient care. And one of the issues with the TNF, which of course has been a really enormously important discovery, primarily focused on rheumatoid arthritis and subsequently on inflammatory bowel disease, discovered in fact by Tiny Maney and Mark Feldman at the Kennedy Institute. Although it works very well for some patients, a number of patients, and in IBD it's something around 20 to 40% of patients will fail to respond. And those that initially fail, what's called a primary non-response, there are also patients who will develop secondary non-responsiveness. And what are the explanations for this? Is this a genetic predisposition? Is the disease too advanced or complicated? For primary non-response, it's not the generation of neutralizing antibodies that can occur within the host and generating secondary non-responsiveness. So we really don't understand what drives the responsive element versus the resistant element. And this is the last part of the studies that I want to mention to you. And we started on this, two very talented fellows in the lab, a clinician scientist, Ahmed Hegatzi, and a basic scientist, Nathan West, were interested in cytokines that were elevated in inflammatory bowel disease in both Crohn's disease and ulcerative colitis that hadn't previously been identified. Were there some things that we were missing? So they undertook a large sort of trawl through the literature with different transcriptomic studies and identified 4 cytokines that were consistently elevated in Crohn's disease and ulcerative colitis. And the highest of those expression level amounts, and also from a statistical standpoint, was this cytokine termed Oncostatin M. Now, when they came to me with this, I have to say I didn't know very much about Oncostatin M and thought, why don't you get on with the actual projects you're meant to be doing rather than trawling around looking for something else that we don't understand. But they were very interested in pursuing this, and all credit to them. OSM is from the IL-6 family of cytokines. It binds to a heterodimeric receptor, including the common IL-6 receptor, GP130, and it has major downstream signaling pathways that involve a number of mediators from STAT3, STAT1, the MAP kinase pathway, as well as PI3 kinase. Interestingly, SNPs in the OSM receptor have been identified in inflammatory bowel disease, GWAS. What about Oncostatin M? Well, Oncostatin M is produced by a variety of different leukocytes. It functions into a number of different systems. It has functions on the immune system as well as a number of tissue cells, but very little is known about the role of Oncostatin M in terms of the gut. But Oncostatin M, the ligand for the Oncostatin M receptor, is also an IBD GWAS hit. So again, another pathway that's being identified by the human genetics. And this is just some of the data from the various transcriptomic analyses that Nathan and Ahmed carried out, showing in this pediatric risk cohort of new onset inflammatory bowel disease patients, and these are children. Oncostatin M was the most statistically significantly highly upregulated cytokine in intestinal inflammation in those patients. We then took this into our own IBD cohort in Oxford to ask the question whether we could reproduce these findings, looking at transcriptomic analysis from our own qPCR analysis of OSM and its receptor in IBD patients that were stratified based on control patients, those with quiescent IBD, mild to moderately active IBD, or severely active IBD. And I think you can see that this pathway is highly upregulated, particularly in patients with severely active inflammatory bowel disease. So did this mean that this pathway may be relevant in patients that do not respond to anti-TNF? So this was the discovery cohort from an Act 1 phase 3 trial of infliximab. These patients are refractory to standard therapy. Colon biopsies were collected prior to anti-TNF therapy. Patients were treated with anti-TNF over this time point, and the response in terms of the clinical response was assessed at week 8. The response was defined as complete endoscopic and histological healing, and what we're showing here is the log2 difference in the expression between resistant patients and responsive patients. And what stood out was this very strong OSM signature in patients that were refractory to treatment with anti-TNF, which had a very striking ROC curve in terms of its specificity and sensitivity in identifying anti-TNF resistant IBD patients. So in another set of patients, these are ulcerative colitis patients, refractory to steroids or immune suppression, then moving on to anti-TNF therapy. Again, the expression of OSM and its receptor was statistically significantly increased in the anti-TNF refractory patients. But it wasn't just OSM and OSM receptor. There was a module of inflammatory cytokines associated with this OSM response that were increased in anti-TNF refractory patients. Indeed, in the patients that did not respond to anti-TNF, 90% of them exhibited this module with elevations of cytokines, chemokines, and integrins. By contrast, patients who responded to anti-TNF, a large proportion of those expressed low amounts of this module, and we've now validated this in a number of cohorts relating to hundreds of patients in both ulcerative colitis and Crohn's disease. So it's relevant in both ulcerative colitis and Crohn's disease. So who is expressing Oncostatin M? Going back to mice and looking at this pathway expressed in different cellular compartments in the intestine, in mice that do have inflammation in black versus those that don't. The production of OSM was most abundant in myeloid cells, and we're just looking at the functionally relevant sources. By contrast, the OSM receptor is expressed primarily amongst mesenchymal cells, activated stromal cells, as well as the endothelium. And in fact, the OSM receptor here, this is just showing its expression on primary human colonic fibroblasts compared to a range of other cytokine receptors. So we had in those studies to identify what were the relevant cell types responding to Oncostatin M. We separated intestinal cells, both tissue cells and immune cells, based on various markers. And here you can see this very nice labeling of the Oncostatin M receptor in the stroma, but not on leukocytes and not on the epithelium. Stimulation of human colonic fibroblasts with Oncostatin M leads to a range of different inflammatory signals, as has been reported. So this is true for intestinal fibroblasts. So it's very different in terms of its signaling than, for example, IL-6 or TNF, and that may be very relevant to how it's modulating intestinal inflammation. If we go into patients and isolate colonic fibroblasts based on the expression of those that express high levels of the Oncostatin M receptor versus those that express low levels, some markers are expressed similarly, but what was striking was that in the OSM receptor high population, there is expression of this module of inflammatory cytokines and chemokines that you find in the patients who are refractory to anti-TNF. And a number of other analyses showed that there's a large accumulation of activated fibroblasts in patients that are refractory to anti-TNF. And this is just showing you, using a marker of activated fibroblasts, podoplanin or GP38, in Crohn's disease, high amounts and accumulation of these types of fibroblasts in the tissue. So finally, coming back from a study in this situation that was started by looking at human samples and hypothesis generating, now we're coming back the other way to ask, is there a functional role? Is there a functional relevance of this pathway in terms of intestinal inflammation? So we're utilizing again our Helicobacter with neutralization of IL-10 model. Remember, I told you that that is refractory to treatment with anti-TNF or indeed anti-IL-6. And mice that are deficient in Oncostatin M have an attenuated form of colitis in this model. It's not completely, it's not that it doesn't develop, but it develops to a lesser extent and resolves more quickly. So this is different to how IL-23 targets the intestinal inflammation. OSM appears to be relevant a little bit later in the response, and taking it away means that the mice recover more quickly than the wild-type mice with this microbe-driven response in the setting of IL-10 deficiency. So OSM then is a previously unrecognized mediator of intestinal inflammation, a potential biomarker of anti-TNF non-response. I think it illustrates an interesting interaction between leukocytes and tissue cells. It constitutes a novel signaling axis between leukocytes and fibroblasts. We're characterizing this further. What other diseases will this be relevant with? But importantly, we believe that this axis is a high-level driver of IBD chronicity and severity, and may be particularly relevant for anti-TNF refractory patients. So if we pull together some of the things that I've been mentioning to you, when there is a relevant microbial driver in the setting of a host landscape where there's a maladaptation, such as deficiencies in IL-10, this leads to the elaboration of a range of innate cytokines, high amounts of IL-23 that can then shape the T-cell response, leading to chronic intestinal inflammation. Alongside that, there's production of Oncostatin M, and we're trying to understand the relevant cellular source of OSM. And when there is elaboration of OSM, this may lead to a different type of pathology to TNF, leading to a remodeling of the tissue in a way that is distinct from TNF, driving accumulation of myeloid cells remodeling the extracellular matrix, and possibly being involved in the fibrotic response. And that's something that we're currently trying to understand. So I want to finish here and suggest to you that as clinicians, I mean, I'm not a clinician, but clinicians tend to think of diseases based on the organs that they affect. And maybe it's time to now start thinking about a redefinition of disease, a new taxonomy of disease, something that my colleague Chris Buckley has spoken a lot about, that is based on not where the disease is manifest, but the processes that are involved. So one of these red boxes could be IL-23. Who are the IL-23 driven? Who are the patients with IL-23 driven disease? Who are the patients with TNF driven disease? Who are the patients with Oncostatin M deficient disease, so that we can target these pathways early in the clinical response, as opposed to patients getting more and more severe and randomly being assigned to one treatment or the next. So exciting times ahead. I think the combination of understanding the leukocyte and tissue cell biology, together with the signals that are coming from genetics, are helping direct our attention towards relevant pathways and trying to understand how to harness them. Not only taking away inflammation, what I haven't been able to talk to you today about is how we might promote those barrier-promoting and regulatory type responses, and probably the combination of those is what will be required over the life course of these complex diseases. So I want to finish here and acknowledge my collaborators and former members in the lab, many who have their own labs now, colleagues in my lab working, following on some of these studies, my colleagues in the translational gastroenterology unit. I want to highlight a collaboration that we had with Janssen here, because when we first identified the OSM signature, we didn't really have enough patients. There wasn't enough transcriptomic data from anti-TNF trials available in the public domain. And actually, I contacted Scott Plevy, who very quickly made a huge amount of data available to us from their in-house anti-TNF trials. And that is a huge resource that I think we should encourage pharma partners to share in these types of studies. And of course, colleagues in Aachen University who helped with OSM receptor fusion proteins, and of course the funding agencies that have supported our work. Thank you.
Wonderful, thank you so much. Time for some questions.
As we discussed, the role of GM-CSF in Crohn's disease is quite complex and probably heterogeneous.
Yeah.
We had a talk earlier today about antibodies to tissue transglutaminase in celiac disease, likely resulting from kind of the proximity issues of the enzyme with respect to gluten. Do you want to, the fact that there are 30% of Crohn's patients who naturally develop antibodies against GM-CSF, do you want to speculate as to mechanisms whereby that might be happening?
Well, I don't really know why they're generating antibodies to GM-CSF. They don't generate antibodies to a lot of other cytokines. I don't know, Judy. We don't really know what the functional relevance of those antibodies are, I suppose.
Right. I mean, it has shown that they're IgG and
that they can neutralize, yeah. So they wouldn't be prolonging the life of GM-CSF, you don't think, in the way that antibodies to IL-2 can, for example.
Hey, Dr. Powrie, really enjoyed your talk. I was wondering if you could comment on the concept that it might be Th1-like cells that have arisen from so-called Th17 cells under states of chronic inflammation and ongoing exposure to IL-23 that might be pathogenic in IV. I guess this is work, supported by work from Casey Weaver's lab.
Yeah, yeah. No, I think that's very possible. They probably started as Th17 cells that progress into a more inflammatory response. Some of the gamma interferon secreting cells, the lineage tracing types of experiments that have been done by Geeta Stockinger and Casey and others would suggest that there is this evolution of the Th17 response away from something that might be more tissue protective to something that becomes more inflammatory. And I would link GM-CSF to that in part, that then renders it more pathogenic, and part of that is to do with sustained expression of the IL-23 receptor. So those Th1's type X Th17 cells that become high gamma secretors still do express the IL-23 receptor.
And a follow-up question, are we not seeing that phenomenon in psoriasis and other disease states that really do respond to anti-IL-17 therapy? I don't know the answer to that. Do you know?
No, I don't know the answer to that, yeah. But I mean, I think even the mouse models would suggest that IL-17 is not a major cytokine driving the inflammatory response in the IL-23 driven intestinal inflammation. Yeah.
Fiona, very nice talk, thank you.
Oh.
I was just wondering if you can comment on whether you have any other data about Oncostatin M with non-anti-TNF modalities. So is it really showing us about severity of disease, or is it showing us specifically about non-response to anti-TNF? Because there must be data out there for ustekinumab or vedolizumab and
There's not
and
so
on.
much that's publicly available, actually, Dermot. You know, when you got into a group of patients that all had severe disease, some patients had a high Oncostatin M module and others didn't, and severity of disease is not a predictor of anti-TNF therapy, so I don't think it's just severity of disease. What OSM, that module, seemed to link to in clinical terms was the need for surgery, and of course that can relate to certain sorts of strictures and types of responses. So my view is that it's actually a different pathological pathway that involves a remodeling of the tissue that could be associated with things like fibrosis.
Yeah, just interestingly, we from surgical resections for Crohn's disease, the unaffected margin, we've discovered that OSM is highly expressed in people who have a shorter time to resurgery in small bowel Crohn's disease. So your last point actually is very, very valid, I think.
So I mean, I think it's important now to understand what that module is driving, what's the cells that are producing that. I think we know something about the responders and how one might, of course, intervene in that, and whether that would be at the level of OSM and its receptor, and there are clinical tools. The trials are waiting to be done. Yeah.
Thank you for a fantastic talk. I was just wondering with respect to cells that are responsive. So if you look in the gut, also we're talking about mate cells, and they're they're much more plentiful maybe than ILCs, and they also express huge amounts of the IL-23 receptor.
Yes.
And I just wondered, do they have a role? Is that to come?
They were on my slide.
Were they? I was
Sort
looking
of
for them.
Unconventional T cells.
Oh,
that
okay. So they're within that
that are
picture.
present, most certainly. And what we see is that, mate cells are difficult to study in mice. So
Okay.
that has made it more difficult to sort of pin down a pathogenic role. They may be playing a more barrier-promoting and sort of host defense role. That would be my guess. And that population, once you start to get chronic inflammation, it becomes diluted by cells that come into the tissue. So those tissue residents it's a generalization, a bit oversimplification, but they may be involved in homeostasis, and then when you bring in these other cells, inflammatory cells, that's what sort of seems to be a view for the tissue Th17 response versus the more pathogenic Th17 response. It might relate to time frames at which cells come into the tissue and how they're conditioned, and the mate cells are there already. Yeah, but we don't know. Yeah.
You suggest that OSM is related to the structuring lesions and so some kind of fibrosis, but this is much more prominent in Crohn's disease and ulcerative colitis, and it seems to me that you show that OSM is increased in both both situations. So how do you interpret it in the context of ulcerative colitis?
It's true. It seems it could relate to the neutrophil response, for example. always just looking at these things. Yeah.
Fiona, there are variants in OSMR that are associated with psoriasis and sclerosing cholangitis as well. Do you have any transcriptional data on patients to find out those tissues that are expressed with OSMR? How does that fit into your kind of biological understanding of what OSMR does?
Yeah, it certainly does. We haven't looked at any other diseases, but everybody's welcome to get on. We can only do so many things, so we haven't looked at that. There is some biology to OSM and the OSM receptor in skin disease, particularly in dermatitis, bit related to an elevated Th2 response. So there's some data about elevations of this pathway related to macrophages and so on. But I think attempts to modulate it haven't been successful in those models. I mean, it wouldn't be fibroblasts from different parts of the body will express the OSM receptor, so it's not difficult to think that that could be a pan mode of communication between leukocytes and tissue-resident mesenchymal cells. So the pathway would be relevant.
So I had a more technical question. So I thought the OSM signature that you show predict response to anti-TNF therapy had an amazing accuracy. So how was that signature obtained? How many genes were in the signature, and what model did you use to get to those accuracies?
Well, I think my colleagues would be better placed to answer that. I mean, this comes from looking at a huge number of different patients, different cohorts, and correlating, using a model to identify the top genes that are associated with the OSM and OSM receptor response.